Hi, and welcome to Lesson 8, Thoroughly Designing a Study, Part E on Intervention Research. And of course, we will start with an overview of intervention research, focusing mainly in this lesson on the data collection and data analysis process. The aim of intervention research is to determine the effects of an intervention. So that could be beneficial effects, but it could also be something like side effects and toxicity. Now, in the data collection, the most typical study design is a randomized controlled trial, but there are many alternative study designs that can be used, which will be discussed in detail during this lesson. Some examples include before and after studies, non-inferiority trials, factorial trials, cluster randomized trials, and propensity matched study designs. Now, in the data analysis, typical outcome measures used include risk measures, survival rates and times, adverse events rates, number needed to treat, patient-reported outcomes such as quality of life scores, and cost effectiveness. And the data analysis varies widely as well, with more simple things like a chi-square test, ANOVA, Student's t-test, Mann-Whitney U-test, to more advanced analyses, including modeling, such as logistic regression models, Cox proportional hazards models, or a simple log-rank test. Now, the reporting guidelines to follow are different than in observational research, since intervention research is intervention research. And so we need to follow the CONSORT guidelines on intervention trials. And there are many specific extensions published based on alternative study designs such as a pilot and feasibility trial, pragmatic trials, non-inferiority trials, within-person randomized controlled trials, and cluster trials. Let's start by putting intervention into the right context. I've already shown you this diagram where proposed mechanisms of pathology are uncovered in animal studies, and usually they first have to be confirmed in clinical etiological cohort studies, but sometimes they go straight to clinical intervention trials if there's a target for example, drug development. And in clinical intervention trials, the efficacy and safety of a therapy needs to be established. Now, the traditional approach to intervention research as follows. The research question is, does experimental intervention improve outcome in domain compared to a control intervention? And the most typical study design is a randomized controlled trial. Now, the data collection goes as follows. Patients with a particular disease are randomly allocated to the experimental study arm receiving the experimental intervention, or the control arm using a one-on-one allocation ratio. And both the patient and the clinicians or researchers are blinded to the group identifier. All patients are followed up after treatment to evaluate whether they develop the outcome of interest. And in the data analysis, a sample size calculation has usually been performed in the study design phase. And the data analysis is usually performed on either a per protocol or intention to treat analysis. And the choice between the two usually depends on the amount of crossover and which analysis shows the most favorable outcome. And the type of analysis itself depends on the primary endpoint. Now, in the conclusion, researchers state that the experimental intervention improves or does not improve outcome in the study population compared to a control intervention. And note that this implies cause and effect relationship. And this is one of the only study designs where you can actually demonstrate that with certainty. Now, you probably already get the sense that this traditional approach is already a lot better than in the other research types. And that is probably due to the fact that intervention research tends to be heavily regulated, especially large drug trials. And these trials oftentimes also involve a statistician or epidemiologist. Now, there are still things to improve though, and we will go into these in detail in this lesson, but for now, very briefly, You should in advance specify in your study protocol whether you are going to use a per protocol analysis or intention to treat analysis. And you should definitely not let that choice be based on which analysis shows the most favorable outcome, but also not the amount of crossover that you will observe. And it is sometimes hard to demonstrate that an experimental intervention does not improve outcome in a study population. Oftentimes it's the case that you're just not able to demonstrate that it does. So that's a very brief summary to the things that are going wrong in this traditional approach, but we'll discuss them in more detail during this lesson. The GCR approach to intervention research is started with the first principle that we have to know the phases of drug research and surgical or complex intervention research. We will start with the well-known drug research phases. Phase 0 is actually preclinical laboratory and animal research, which will not be discussed during this lesson. Phase 1 is a safety study, and usually it's done through a prospective dose escalation study in a few volunteers or patients. And the goal of this study is to find the maximum tolerable dose. And so this is a study that is heavily driven on safety. Sometimes efficacy measures are measured, but they're not the goal of this study. In the phase 2 study, we want to evaluate preliminary efficacy. And usually a prospective cohort study is done in more patients than during the phase 1 study. And the goal here is to evaluate whether there seems to be some efficacy. And the second part of the goal of this study is to gather some more data on the safety profile of the intervention. Now, we also use the estimated efficacy for sample size calculations to design a phase 3 trial, which is a comparative efficacy study. And it's usually in the form of a randomized controlled trial in many patients. Here the goal is to evaluate whether there's a benefit in efficacy and or safety over standard care. And then if that is demonstrated, then usually the next step is the evaluation of market approval by the FDA. But there's also a fourth phase that is called the post-marketing study. And it's usually a registry database study in many patients. And the goal here is to evaluate long-term benefits and side effects. You might already be asking yourself the question, what about placebos? Well, here it comes. Do you need a placebo control? If you use a placebo control, then we call that trial an explanatory trial, because you're trying to prove, or in other words, explain the biological efficacy of a drug outside the placebo mechanism. And that is actually important to the pharmaceutical industry. So if you don't use a placebo control, then that trial is called a pragmatic trial. And the thought behind this is that since the placebo mechanism is actually always present in a therapy, you just take a pragmatic approach and do not try to control for it. The interesting control in these kind of trials is actually the standard of care as a comparator. And this is important to the researcher and patient because we try to move forward in clinical practice. So you always want to know whether an experimental therapy performs better than an already established therapy. The only other case where it makes sense to use a placebo is if there is no established therapy for a disease. Now, if we look at these drug research phases, then it would make sense if you want to perform a placebo randomized controlled trial to perform that explanatory trial in phase 2. And the pragmatic trial is supposed to be in phase 3 because we want to evaluate whether there's a benefit over standard care. Now, the former research phases apply to drug treatments. Surgical therapies or other complex interventional therapies require some special considerations. And so the ideal recommendations have been published as a guide for the research phases specifically for surgical and complex intervention trials. And IDEAL stands for Idea, Development, Exploration, Assessment, and Long-Term Follow-up for the improvement of the quality of research in surgery. Now, what are the differences between drug and surgery research? And note that I'm saying surgery, but something like an interventional radiology procedure or other complex intervention is meant to be included in this term. Now, in drug research, the majority of the developmental costs, like time and finance, is preclinical, whereas in surgical or intervention research, the majority of the developmental costs is early clinical. Now, in drug research, the optimal dosing strategy is the bottleneck, whereas finding the best way to do the procedure is the major bottleneck in surgical research. And in drug research with adequate dose found, the therapy can be highly standardized and predictable. Whereas in surgery, there is almost an endless possibility to vary the procedure with a long way to evidence of superiority. So therefore, the procedure is very hard to standardize. In drug research, there is no operator dependency and no learning curve. Whereas in surgical research, most of the time, the therapy is highly operator dependent and there is an important learning curve to take into consideration. The first phase in the IDEAL recommendations is phase 0, which is actually the pre-IDEAL stage, where preclinical research is performed to define the procedure and determine its feasibility. And the goal here is to avoid predictable risk of failure or harm to the first human patients. Stage 1 is the idea first in human use stage, where structured case reports are published that provide full details on the innovative procedures. And the goal here is to provide a proof of concept and to define its clinical relevance. And it's important to also describe what doesn't work. So avoid only reporting success cases. Now, stage 2 can be subdivided in different parts. The first part, stage 2A, is the development stage, where prospective development studies by innovators and only early adopters are performed to develop the intervention to a state of stability sufficient for replication by others. So that means that all problems need to be solved and the procedure needs to be refined, further optimized until it can also be done by others than the first experts. Once this has been established, the procedure can go into the next stage, stage 2B, the exploration stage, in which prospective studies, and these can be multi-center exploration cohort studies or a pilot multi-center randomized controlled trial, further address questions in the preparation for stage 3. And so there is some kind of overlap between the developmental stage and the exploration stage. But in the development stage, it's usually only the early adopters and innovators that perform the procedure, and in the exploration stage, it's now also in the hands of other operators to perform the procedure. And in this stage, it should be demonstrated that the treatment can be performed in a standardized fashion with beneficial outcome. Once that has been achieved, the procedure can proceed to the next stage. And stage 3 is the assessment stage, similar to drug trials, where randomized controlled trials evaluate whether there's a benefit in efficacy and or safety over standard care. And phase 4 is a long-term study stage, also similar to drug trials, where registry database studies in many patients identify long-term rare adverse effects, changes in indication or delivery quality over time. Now, we had already learned that stage 2 is the crucial stage here in surgery and complex interventions, which differs significantly from the drug development stages. And actually, a new surgical or complex intervention procedure can get stuck in stage 2 for years until it has been optimized. And that's okay, because if it gets pushed too soon into phase 3, then an unfair comparison with standard care is performed because the procedure is not yet optimized. And that might significantly diminish hope with regards to this new treatment, especially considering the time and financial investments that are necessary to perform a randomized controlled trial on a surgical intervention. The second principle of the GCR approach to intervention research is to use your study goal to choose between different types of trial designs. And let's look at a list of options with regards to the study design. You can use a randomized trial or a non-randomized trial. You can use an explanatory, so a placebo-controlled study, or a pragmatic trial, blinded or an open-label trial, a classical parallel group or factorial design trial, a superiority or a non-inferiority trial, a within-patient study, before-after study or crossover randomized controlled trial. You can use an adaptive trial design, and things like a cluster randomized trial or multiple cohort randomized controlled trial. And we will discuss these study designs in detail now. Let's start with randomization. Because randomization is such a strong method to counteract bias by confounding, and thus randomization actually enables inference of causality, I think you should use a randomized trial design, except for if you want to run an early phase trial, so for example, drug phase or ideal stage 1 or 2, or if it's really not feasible to perform a randomized trial. But I would personally not recommend trying to infer causality from a non-randomized study with a matched study design, such as the propensity score method. And if a randomized trial is not possible, I think it's actually purer to describe the two separate cohorts and see what you can learn about the benefits and disadvantages of the two separate treatments. Now, blinding and using a placebo control are related but not the same. So in explanatory trials where we use a placebo to prove biological mechanism, and by the way, in surgery that's called a sham study, at least the patient is always blinded. And most of the time the treating physician is also blinded. But of course, in surgery, the operator still needs to know which procedure to perform. And ideally, the observer that assesses the outcome under study should also be blinded. But in pragmatic trials, it differs between drug trials and other types of trials. In drug trials, there's also often a triple blind design where no one knows which drug is given to which patient. In contrast, pragmatic intervention and strategy trials are usually open label. So, who is blinded in studies? Well, the outcome assessor should always be blinded if possible. And if it's only the outcome assessor, you may call this an open label design, and more specifically a probe design, a prospective randomized open blinded endpoint design. And if the outcome assessor is not blinded, observer bias may cause problems, especially with soft endpoints. Now, if the patient, the caregivers, and the outcome assessor are blinded, we call this a triple blind study design. And we do this to prevent observational selection and observer bias. Now that's it on placebos and blinding. Let's move on to the classical design for a randomized controlled trial, a parallel group trial. In such a design, eligible patients are selected and included in the study and subsequently randomized to two different treatment arms, and usually 1-to-1 allocation ratio. So there's a 50% chance of being assigned to experimental therapy, or control therapy. And after having received these therapies, patients are followed up, and the difference in outcome is assessed between these groups. Now, in a factorial design, basically another layer of comparison is added. We start again with selecting eligible patients and including them in the study, and then first randomize for factor 1, so patients get treatment in arm A or treatment in arm B, but then we subsequently randomize for another factor, number 1 or 2, so patients can get treatment in arm A with factor 1, arm A with factor 2, arm B with factor 1, and arm B with factor 2. And this might be something like a combination of drugs, or the second factor might be a difference in dosing. And in this way, multiple comparisons can be made in one randomized controlled trial. Now, patients receive their respective therapy, are followed up, and then we can draw multiple comparisons of outcome. Now, assuming that the components don't affect each other, so there's no significant interaction between the treatment arm and these two factors, we can, in the group that received factor 1, compare treatment arm A to treatment arm B, and do the same for the group of patients that received factor 2. And in the group of patients that is assigned to treatment arm A, we can compare factor 1 to factor 2, and do the same for the patients that are assigned to treatment group B. So basically, we can perform 2 randomized controlled trials in one without significant increase in the sample size. Now let's move on to superiority and non-inferiority trial. Most clinical trials are designed as a superiority trial, where the goal is to demonstrate that a new therapy is more efficacious or less toxic than the comparator therapy. And we can have different scenarios in such a trial. Let's start with a positive trial result. First, we perform a sample size calculation where we have to estimate the difference in efficacy between the therapies. And let's say we estimate that there will be a clinically meaningful difference, and we state the power and the alpha that we want to use. Then we perform the study, and we observe a large estimated difference between the treatment groups. Now, in our analysis, we construct a narrow 95% confidence interval around the estimated difference, with the lower limit of the 95% confidence interval being larger than the equality measure, and we find a small p-value of the primary statistical test. Now, the interpretation is that the observed data are highly incompatible with our null hypothesis of there is no difference. And thus, we assume that the alternative hypothesis that there is a real difference is much more likely. Now, in a negative or indeterminate trial result, we start with the same sample size calculation, but now we only observe a small or even no difference. And this could be either way. And eventually we find a wide 95% confidence interval, which includes the measure of equality. And there's a relatively large p-value of the primary statistical test. The interpretation of these results is that the observed data are not incompatible with the null hypothesis. So there might actually be no difference. But note that we haven't proven the null hypothesis of no difference. Some trials are actually designed as a non-inferiority trial, where the goal is to demonstrate that a new therapy is at least as effective as the comparator treatment. And the largest accepted efficacy difference needs to be clearly defined in such a study. Usually the new therapy has another advantage, like being less toxic or invasive, less expensive, or having a wider availability. And for these kind of studies, we need to use the CONSORT extension on non-inferiority trials. Now let's look at scenario 1 of a positive trial result. We start again with a sample size calculation, but here we need to include the clinically acceptable difference, which means that if the trial ends up showing a difference that is smaller, then we consider the treatments equally effective. But if the trial ends up showing a difference that is larger than the clinically acceptable difference, then we cannot state that there is non-inferiority. And of course, we also need to indicate the power and the alpha level that we want to use. And let's say in our study we observe a very small estimated difference between the two treatments. And in our analysis, we get a very narrow 95% confidence interval on that estimated difference, with the upper limit not reaching the predefined acceptable difference, which means that the difference as estimated by the 95% confidence interval is actually smaller than the predefined acceptable difference. And thus, our interpretation is that if there is a difference, it is not likely to exceed the clinically acceptable range. And because a non-inferiority trial is powered on a small difference, it usually requires a larger sample size than a superiority trial. And a thing to add is that, of course, that small estimated difference can be in either way, but if you have demonstrated non-inferiority, you can usually proceed to test for superiority of the experimental therapy in terms of efficacy and toxicity at no statistical cost, because Now, let's look at scenario 2, a negative trial result. We start again with the same sample size calculation stating the clinically acceptable difference, but now we observe a small to relatively large estimated difference between the therapy efficacies. And in the analysis, we get a wider 95% confidence interval with the upper limit including the predefined acceptable difference, which means that the difference in treatment efficacy can be larger than the acceptable difference. And the interpretation is that there may be a difference exceeding the clinically acceptable range, and thus we cannot demonstrate non-inferiority in the study. There are different other designs like a within patient before and after or crossover RCT, and in all these designs we use the patients themselves as their own control. One example is the use of a split body design. And here for each patient we randomize the therapy into experimental and control for 2 different body parts. So a patient could end up with the assignment of body part 1 to the experimental therapy and body part 2 to the control therapy, or the reverse. And eventually we assess the outcome in both body parts, and then we pool the outcome over all the patients for the body parts assigned to the control therapy and the body parts assigned to the experimental therapy. And that's how we can compare the pooled outcomes of control and experimental therapies. Now we can also use a before and after or so-called crossover design, in which each patient is randomized to receive the experimental or the control therapy upfront, and then we wait until patients can develop an outcome, and then we wait a little longer for a certain washout period so that we make sure that the effect of that upfront therapy is not any longer in the system. And then we give each patient the other therapy. So in this case, the patients that got experimental therapy up front now get the control therapy, and the vice versa is true for the patients that up front received the control therapy, they now get the experimental therapy. And then we wait again until patients can develop an outcome. And here we pool the outcomes of the control therapy moments and the experimental moments to compare the effect of those therapies. And for these studies, you need to use the within-person randomized trials extension to the CONSORT guidelines. Now, let's focus a little bit more on that split body design. This can be applied in multiple diseased body parts that can be studied separately, so it would make sense for example, the eyes or ears or knees and teeth. And the benefit is that it's a really efficient design, and you actually eliminate all the between patient variability that you would get with a standard randomized controlled trial. And it's well suited for technique development studies, like a surgical technique. But you should be aware of certain things. So there should absolutely be no significant carry across effect. The treatment of one body part should not affect the other body part. And systemic toxicity differences cannot be evaluated separately. And of course, because these are clustered or dependent data, they require a more complex analysis. And if we focus on the before-after crossover design, we can state that this design can be used to study the therapeutic efficacy in, for example, chronic non-curable disease. And the benefits here as well are that it's an efficient design that eliminates between patient variability, and it is particularly well suited for patient-reported outcomes. But be aware that drug trials should be blinded, and they can be performed placebo-controlled, and you need an adequate washout period to prevent any carryover effects. And also here, it requires clustered paired data analysis. Now, the next form of study design to be discussed are the cluster randomized controlled trial designs. And for changes in complex diagnostic, prognostic, or therapeutic strategies, or policies and educational projects, it makes more sense for research to take place on a group level instead of individual patient level. And so in a classic cluster randomized controlled trial, entire groups of patients, which are referred to as clusters, are simultaneously randomized at baseline to the current or a new approach. And a cluster can take various forms, like a general practitioner's practice, a hospital, a group of hospitals, etc. And for these kind of studies, we should use the cluster randomized trials extension to the CONSORT guidelines. Now, let's imagine that we identify multiple hospitals as eligible to take part in our cluster randomized controlled trial. Now, those hospitals themselves are clusters, and we randomize them to either a new strategy or a control strategy. And then we measure outcome across these hospitals to compare the efficacy of the new strategy to the control strategy. And as a rule of thumb, each arm should at least consist of 4 clusters. And the multi-level hierarchy, so from clusters to individual patients, requires statistical methods that incorporate the paired nature of the data. And this can take the form of, for example, a generalized linear or nonlinear mixed effects regression model, or something like a general estimation equation model, which will not be discussed further in this course since these are pretty advanced statistical techniques. Another form is to use a step-wedged cluster randomized controlled trial, where we randomize the clusters to a time point in which they start to implement the new strategy. And you can see here that the first cluster keeps using the standard strategy at the start of the study until the study is almost over, and then they switch to the new strategy. Whereas down the line of the clusters start to use that new strategy earlier on. And we can use this as a sort of cluster randomized version of the crossover trial to assess the efficacy of the new strategy compared to the standard strategy. And it should be noted that we should use the step-wedged cluster randomized trials extension to the CONSORT guidelines. Now, cohort multiple randomized controlled trials are yet another form of a trial design. We start with an observational prospective cohort study in which we include patients with a certain disease. And we keep extending that cohort over time and follow each patient up. And all patients give informed consent for that cohort study. And now, during the cohort study, we start to design little trials. So for example, patients that would be eligible to a new intervention called A are split up in a group where we select random patients that are asked for an informed consent to receive this new therapy A, and the other patients are not asked to receive that therapy A. So they basically function as a control group. And then we can do the same for multiple other therapies and thus start multiple randomized controlled trials within the prospective cohort study. And during the prospective cohort study, we standardize outcome measures at fixed moments in the follow-up. And at those moments, the experimental intervention groups are compared to the control groups. All right, now that's the basics of trial designs. Now let's move on with the third GCR approach principle, and that is to predict potential sources of bias and think of ways to avoid them. And again, we have extensively discussed this in the formal lesson, so I'll sort of fly through it. But selection bias, information bias, and confounding play a role in intervention studies, and we have different remedies for it. And blinding is an important one to prevent information bias, whereas randomization is an important one to prevent confounding. But if we randomize, then we also should use an intention to treat analysis. In addition, we can use multivariable regression modeling on the primary endpoint and or stratified analysis. And of course, we can cautiously limit our conclusions to the population that we actually studied. Now, the fourth principle of the GCR approach is to pre-specify your main analysis type with its rationale, an intention to treat versus the per protocol analysis. And if we compare them, and we see that the intention to treat analysis is the type of analysis where all patients are included in the treatment group as randomly allocated, regardless of the therapy that they actually received. Because sometimes patients drop out or they cross over to the other treatment arm. But the strength of this approach is that it maintains group comparability as per randomization. The weakness is that it does not compare the effects of the actual treatment received, and in the case of crossover from the control to experimental therapy, the therapy effect may be diluted. The per protocol analysis is the type of analysis where all patients are included in the treatment group as actually treated, regardless of the random allocation. And so it compares the effects of the actual treatment received. But it may introduce bias due to group incomparability because we have broken the randomization. And so the intention to treat analysis should absolutely be your standard go-to analysis. And the per protocol analysis should not be used too easily. However, there is a place for the intention to treat analysis plus a per protocol analysis in non-inferiority trials. And you should definitely state in your study plan in advance which one of two analysis types is your primary one. And you shouldn't wait until you see the results so that you might be biased to choose the one that suits you best. Now, the fifth principle is to pre-specify your hierarchy of study endpoints. You should include in your predefined statistical analysis plan what your primary endpoint is, and this is what the study is aimed and powered to test, so it has the strongest evidence. And an example for this could be median overall survival. You should also specify secondary endpoints that provide important additional information about the treatment. for example, complication or toxicity rate. But here the strength of evidence has already decreased. And the third layer is to include potential subgroup analyses or interactions that are tested. And these should definitely always be pre-specified and not post hoc, and regarded as nothing more than exploratory. Primary and secondary endpoints. Choose your primary endpoint wisely and make sure it matters clinically. And if you use composite endpoints, pre-specify them and meticulously define them. And remember that the most frequent component will drive the event rate. And you shouldn't mix efficacy and safety endpoints in a composite endpoint. Use separate endpoints for them. And in relatively large trials, it is not necessary to adjust for covariates in the analysis. So you don't necessarily have to use a multivariable model to assess the primary endpoint. You can do it just to increase the power, but if you do that, pre-specify it in your analysis plan. With regards to reporting your endpoints, you should at least report these items. In uncensored outcomes, such as risk and rate outcomes, you should specify the number of events observed in the different groups, as well as the absolute risk difference, relative risk, and, if interesting, the number needed to treat, together with the 95% confidence intervals. Now, in censored outcomes, you should mention the number of events and number censored in the different groups, as well as the odds ratio for logistic regression and its 95% confidence interval. In time to event outcomes, you should also mention the number of events and number of censored in the different groups, together with the median time to event difference and the hazard ratio for a Cox proportional hazards regression, along with its 95% confidence interval. So the p-values are the least important, but if you mention them, use exact p-values instead of cutoffs. And with regards to subgroup analyses, if you deem them necessary, then limit your subgroup analyses and pre-specify them in your analysis plan. Now, it is better to test for interactions in the main analysis instead of simply testing for subgroup differences, and most statistical software packages let you do that quite easily. You create a new variable that is actually the product of two already existing variables of which you want to see whether there is an interaction between them. And then you can enter this newly constructed product variable into the model. And if you add this to your model and it is significantly associated with the outcome, then that indicates an interaction. Interactions can be graphically displayed in a forest plot quite nicely. And as previously stated, subgroup analyses should be considered exploratory in nature, so don't overstate the importance of subgroup analyses. Now, the sixth principle of the GCR approach is to perform the sample size calculation to your best ability. And you can use the online GCR statistics sample size calculator that I already showed to you to do this. And required ingredients for simple sample size calculations are the estimated treatment effect, and small treatment effects usually require large sample sizes. Power, a conventional number is 0.80, an alternative number is 0.90, which will require a larger sample size. And alpha level, which is conventional at 0.05. 0.01 will require a larger sample size, and specify whether you want to use a two-tailed test or a one-tailed test, and if necessary, incorporate your expected dropout or loss to follow-up rate in the sample size number calculated. Now, this is a screenshot of how the sample size calculator on the GCR statistics website looks like. You can readily use it to calculate sample sizes for the comparison of proportions, comparison of means, and for correlations and regressions. Now, more complex sample size calculations on non-parametric tests, logistic regression analysis, survival analysis, tests of agreement and reliability, as well as sequential or interim analysis design trials, require other software packages, and it might be wise to consult a statistician. The seventh principle of the GCR approach is to use a robust method of randomization. The workflow in a trial goes as follows. You want to screen an eligible patient, and if that patient can be included in the study and provides informed consent, then that patient gets randomized. And the essence of that randomization is that it may not be influenced, it should be irreversible, and it should be logged. And after that randomization, the patient is allocated to a treatment. Now, there are different ways to randomize, and in terms of an operational aspect, the preference nowadays is to use a computer-generated randomization, and preferably on a safe online server. And the allocation ratio can be balanced, which is basically 1:1, or unbalanced 1:2 or 1:3. And with regards to the types of randomization, we can subdivide simple randomization, a stratified random permuted block randomization with varying block sizes, and minimization. In simple randomization, the computer basically performs a coin toss of randomly either allocating a patient to treatment arm A or treatment arm B. Permuted block randomization allows researchers to have a little bit more certainty about the balance of the treatment groups, because here the computer uses a pre-specified but secret sequence of allocation. So for example, AABB, AABB, to assign patients to the different treatment arms. Now, the benefit of this is that you can add strata where those permuted blocks are different. And so you can make sure that, for example, in a multi-center randomized controlled trial, the baseline characteristics over the different hospitals are comparable. Now, the essential thing, though, here is that you need to vary the block sizes. So if your first block, for example, is AABB, AABB, then the next block shouldn't have 6 allocations as well, but should, for example, only have 2 or 4. So it could be something like BAAB. If you don't do that and you use the same block sizes, then eventually you will be able to tell to which arm a next patient will be allocated. And that's something that must be prevented. Now, minimization is even more extreme, because this actually is a type of modeling that assigns patients to treatment arms with a different weight based on the current distribution of baseline characteristics in the treatment arms. And so you use a model to sort of make sure that patient characteristics will be evenly distributed between the treatment arms. Now, the permuted block randomization and minimization may have some additional benefits, especially in relatively smaller randomized controlled trials. But if you have a nice, large randomized controlled trial, then simple randomization should do the trick. Now, one thing that I find very annoying is that I see a lot of randomized controlled trials give you a baseline table with baseline characteristics in the experimental group and the control group, and then a univariable test to see whether there are significant differences in the values of those baseline characteristics. In my mind, this makes no sense at all, And there are also journals that will prohibit this. So I would say don't test for those differences. Because if you find no difference, absence of significance does not equal equality. And small differences will become significant in very large trials because those simple statistical tests that will give you that p-value are heavily driven by sample size, since larger trials tend to have smaller standard errors, and the standard error is incorporated into the calculation of the p-value. And most of all, the difference that you will find will be due to chance if the randomization was adequate and your trial is large enough. The eighth and last principle of the GCR approach to intervention research is to become familiar with the role of a data monitoring committee. So, with regards to the terminology here, the sponsor of a trial is an individual, institution, or a company taking responsibility to initiate, manage, and finance a clinical trial. The investigator is the one actually conducting the investigation. And sometimes that's the same as the sponsor. And that investigator can also be an individual or, for example, a hospital. And the Data Monitoring Committee is an independent committee consisting of physicians and a statistician that confidentially reviews unblinded trial data at interim analyses and can recommend stopping or altering the trial to the sponsors and investigator. And the tasks of the Data Monitoring Committee is to evaluate potential harm to study participants based on the recorded adverse events rates. And the investigator needs to continually record adverse events in trial participants, and those records can be reviewed by the DMC. Another task is to detect overwhelming evidence for superiority in the experimental arm, and the last task is to detect futility of the experimental therapy. In all these three cases, the Data Monitoring Committee will evaluate whether it would be best to terminate the trial early, because in the first situation they do not want to pose additional patients to the same harms. In the second situation, they don't want people to get an inferior treatment, so the analysis for this trial should be done as soon as possible, and the findings should be communicated as early as possible. And in the last case, it wouldn't make sense to keep on giving patients the experimental therapy while it is clear that the control therapy outperforms the experimental therapy. Well, that's it on intervention research, and great job again hanging in there. I'd say get ready for the last part on thoroughly designing a study, reproducibility research.